The answers when you come back. Let's answer the questions that we were just discussing. Does this gel show, are these people my parents? Can these people be my parents? Well, the answer to can these people be my parents is yes. The reason that that's true is that, as you can see, the person I refer to as Dad only has one length of DNA, the 22-letter long version. If this were my dad, he would have to give me the 22-letter long version, and I have that version. I also have a version that's 18 letters long, but so does my mother, so I could have gotten the 18-letter long version from my mother. Therefore, this gel is consistent with these people being my parents, i.e., these two people each giving me half of their DNA. Well, are these people my parents? This gel does not prove that these people are my parents, although the gel is consistent with them being my parents. The reason the gel doesn't prove they're my parents is that if every person only has one of three possible patterns that they can show in this gel, either only 22 letters long, only 18 letters long, or both 18 and 22 letters long, then given that there's 7 billion people on Earth, you could pick any two people at random and they might be consistent with me in this gel. However, this is actually how we do paternity testing and forensics. So how do we use this technique for paternity testing and forensics if this gel doesn't prove that these people are my parents? Well, the answer is that you have to remember that we are just looking here at a very small number of letters from a total number of letters of DNA that's 3 billion letters for my mom and 3 billion letters for my dad. If we ran another gel and we looked at another segment and these people were consistent with me, you would be more convinced that they were my parents. If we ran an infinite number of gels looking at many different parts of the DNA and they always matched me, you would be very convinced these people were my parents. So you can actually ask a statistician, how many regions of the genome do you have to look at and have these people match me so that you can say that they most likely are my parents given that there's 7 billion people on Earth. And statisticians can answer that question and tell us that we have to look at about 20 different sites on the DNA. So that's how this technique works. And now I'd like to do an example with you. First, we're going to do an example with paternity testing, and then we're going to do an example with forensics of how to use this technique. So on my next slide, I'm going to show you the introduction to this exercise. The first example is that there's been a mix-up in a maternity ward. And there's 3 babies, A, B, and C, and 3 sets of parents, 1, 2, and 3. And we're going to match which sets of parents go with which baby. So on your worksheet, you'll see a similar picture to the one I'm showing on my next slide. Here's a region of chromosome 15. This region has a repeat of TTA, GGAT in the middle. And this region can be repeated n number of times, so a different number of times in different people. We're going to analyze this region of chromosome 15 in the babies and the parents to see if it can tell us which babies go with which parents. On your worksheet, you'll also see the following picture. This is the result of the DNA analysis of chromosome 15. You can see the ladder markings on the left, and you can see the patterns that are given by all of the parents and all of the babies. So what I'd like you to do is answer the questions on your worksheet, and those questions are shown here. First, given the data so far, which of the 3 babies can you conclusively connect to a set of parents? Second, how did you conclude this? Third, why can you not determine at this point all of the babies and all of their parents? And finally, how do you think you would go about conclusively determining all the matches of the parents to the babies using DNA fingerprinting analysis? Let's go through the answers to the questions on your worksheet. First, the baby who you can conclusively connect with a set of parents is baby A, who goes with parents set 3. The way you can conclude this is the 25 length repeat. So if you look at your ladder and you find n equals 25, if you see that band is only present in dad number 3 and in baby A. Baby A had to have gotten the 25 repeat length from someone, and the only person they could have got it from is dad number 3. That's how you can conclude that dad number 3, and therefore also mom number 3, goes with baby A. The other 2 parents, 1 and 2, actually cannot be matched at this point with babies B or C. And that's because if you look at the types of genes, the genotype of parents number 1 and parents number 2, and you think about all the different possible genotypes of children that they could have, they could actually each have either child B or C. So how would we actually go about concluding whether parents 1 and 2 go with baby B and C? Well, remember, this is just a small segment of chromosome number 15. These are maybe hundreds or thousands of letters of DNA, but we have 3 billion letters of DNA from our mom and 3 billion letters of DNA from our dad. So we can go look at another region of another chromosome and see if that one answers our question. So now if you look at your worksheet, you'll see a picture like this. This is a region of chromosome 4, and in this region, the sequence CAG is repeated a certain number of times in the middle of the sequence. So now I'm going to show you another gel, also shown on your worksheet, that shows the results of analyzing these same people at a different region, a region of chromosome 4. What I'd like you to do now is analyze this gel and answer the questions you see on your worksheet. Those questions are shown here. Given all the data in this problem, I want you to match the 3 sets of parents with the 3 babies. Then I also want you to explain how this site on chromosome 4 allowed you to match parents 1 and 2 with babies B and C. Let's go over the answers to those questions. The first question is to conclusively connect all 3 sets of parents with all 3 babies. Well, we already know from chromosome 15 that baby A goes with parents 3. So what we can find from this gel is that baby B goes with parents 1 and that baby C goes with parents 2. So how do we find that? If you look at this gel here, baby B has 2 copies of the 80 repeat length. If you look at parents 1 and parents 2, both dad number 1 and mom number 1 have that 80 repeat length. Thus, they could have each given the 80 repeats to baby B. However, neither dad number 2 nor mom number 2 have that 80 repeat length. So parents number 2 could not be the parents of baby B. If you look at baby C has the repeat length of 70 and the repeat length of 40. If you look at mom number 2 is the only parent who has the repeat length of 40, and thus she could have given this 40 repeat to baby C. Baby C's other repeat is 70 long, and dad number 2 has the 70 repeat length, so he could have given it to baby C. Thus, in conclusion, parents 3 go with baby A, parents 1 go with baby B, and parents 2 go with baby C. So that's how you match the babies to the sets of parents. Now, this technique of DNA fingerprinting, as we've discussed in this example, can be used to identify people for paternity testing. But this same technique can also be used in forensics analysis, and I'd like to show you an example of that now. Do you remember the gel that we were looking at earlier in the lesson in lab? Well, I'd like to show you that gel again. In this gel, you can see that lane 5 contains the ladder, and lanes 6, 7, and 8 contain the DNA of 3 suspects. We'll call them D, E, and F. Lane 9 contains DNA found at the crime scene. What I'd like you to do is analyze this gel and answer the following question. Which suspect, D, E, or F, has DNA that matches the DNA from the crime scene? So in the gel you just looked at, suspects D, E, and F had the following DNA patterns. Suspect D had 1 band that was higher up in the gel. E had 2 bands, one that was higher and one that was lower. And F had 1 band that was lower. When you looked at the DNA from the crime scene, there was 1 band that was lower, and it was the same length as person F. Thus, suspect F's DNA matched the DNA found at the crime scene. That's how DNA fingerprinting analysis can be used in forensics. During the filming of this lesson, I had the opportunity to visit the identification unit at the Cambridge Police Department. This is the lab where the Cambridge Police Department can investigate items found at a crime scene for things like fingerprints or for DNA. So for instance, here you can see that I'm getting my cheek swabbed. This is how the technicians at the identification unit isolate DNA from someone who's potentially a suspect. They can also isolate DNA from items found at a crime scene, such as a water bottle or a weapon. You can watch a full video tour of the identification unit in the video that's found after this lesson. I hope that you've enjoyed the lesson today as we learned about DNA fingerprinting analysis, how it can be used to identify people based on their DNA, and how it works, as you saw in my lab. Thanks for joining me, and I hope you enjoyed learning about paternity testing, forensics analysis, and DNA fingerprinting. Thanks for your interest in my Blossoms lesson on using DNA fingerprinting in paternity testing and forensics analysis. The main objectives of this lesson are for students to learn how DNA fingerprinting works, to see it actually work as a technique in lab, and to be able to analyze the data that is generated by DNA fingerprinting and use it in situations such as paternity testing and forensics analysis. There's no background information necessary for this lesson, although it's helpful if students already know that DNA is the genetic material and is made up of A's, T's, G's, and C's. And it's also helpful if they know that the genetic material of any one person comes from their parents, and that 50% of that material comes from the mom and 50% comes from the dad. There's no necessary materials for the lesson other than the worksheets that are provided and an ability to project or print out the slides. There's 9 segments to this lesson, and I'll go through each one and what happens in each segment and between segments now. In segment number 1, I introduce the concept of how you look at a DNA sequence, a series of A's, T's, G's, and C's. And I ask the students that if you were looking at 2,000 letters of DNA, how many of those letters might be different between any 2 people on Earth? I give the students time to brainstorm this number, and usually students guess a lot higher than what it actually is, which is 1 or 2 letters. So when I come back in segment 2, I reveal that only 1 or 2 letters out of 2,000 would be different if you compared any 2 humans, because any 2 humans are 99.9% identical. Then I talk about how the differences in DNA, those less than 0.1% of letters, can be used to tell people apart, and I ask the students to brainstorm uses for a technique where you would be able to tell people apart based on their DNA. So after segment 2, most likely the students may come up with either paternity testing or forensics. But in segment 3, I come back, go over those uses, but I also talk about how at the Broad Institute where I work, we use this technique all the time to find genes that are associated with disease. Then in segment 3, I introduce how DNA fingerprinting can work to look at the differences in length of DNA between people, for instance, a length of a repeated sequence between 2 people, as opposed to differences in the actual sequence or what the letters are. These are the differences, these length differences, that are usually used in paternity and forensics analysis. So I talk about how you can separate pieces of DNA by size in gel electrophoresis, and I pose 2 questions to the students about how they would get DNA to move through a matrix, given that DNA is negative, and also what they think would happen to the speed of DNA if you raced a big piece of DNA versus a small piece of DNA through a matrix. When I come back in segment 4, we talk about how you can get DNA through a matrix by making the other end of the matrix positive so that the DNA is attracted to it, and how large pieces of DNA move slowly through the matrix and small pieces move quickly. Then I introduce the concept of what this technique would actually look like. And then in segment 4, we actually go into the lab. I do a DNA fingerprinting experiment, and I show the results of a gel at the end, where there are 3 different DNA samples. The DNA samples, one has a large piece of DNA, one has a small piece of DNA, and one has 2 different pieces of DNA, a large and a small. So some people show 1 band in the gel and other people show 2 bands. And I asked the students to write notes about how many bands they saw in each lane, whether the bands were longer or shorter in each lane, but also why it is that some people show 1 band and other people show 2 bands. We answer this question in segment number 5, and the answer is because every person gets half their DNA from their mom and half their DNA from their dad. And if the mother and father's DNA is of different lengths, then the person will show 2 bands. But if the person inherits the same length from both parents, the person will show 1 band. This is usually a very difficult concept for the students to grasp. And so I go on to show this concept as a picture where I show the nucleus of a human cell. I show the maternal and paternal versions of 3 different chromosomes and label one section so that they could see that that little section might be slightly different lengths because the maternal version and the paternal version had a different number of repeats. Then I show the students a sample gel with DNA from 3 people. A person I label as me, a person I label as the person I refer to as mom, and then a person I refer to as dad. And I asked the students if the gel shows whether or not these people can be my parents and whether or not these people are my parents. In segment 6, I explained that the 2 people can be my parents because the gel is consistent with my DNA, but that it doesn't prove it. And so I talk about how we use many regions of the DNA to actually show whether we think that someone is the parent of a child or we think that someone's DNA has been found at a crime scene. Then we start the main exercise of the lesson, where I give the students a gel showing 3 babies and 3 parents, and asking them to use the DNA in this gel to figure out which set of parents, 1, 2, or 3, goes with which baby, A, B, or C. When we come back in segment 7, we discover that you can tell that baby A goes with parents 3, but you actually can't tell which of the other 2 sets of parents go with the other 2 babies. So then we go on to examine another site on a different chromosome that actually will tell us which parents, 1 or 2, go with which baby, B or C. The students analyze that gel from that other region of a different chromosome between segments 7 and 8, and then in 8 we go over the final answers, which are that baby A goes with parents 3, baby B goes with parents 1, and baby C goes with parents 2. Finally, I show the students an example of a gel from forensic analysis, which is actually the gel I ran in lab. In this gel, there were 3 suspects, D, E, and F. D had the 1 band that was higher in the gel, E had 2 bands, 1 higher, 1 lower, and F had 1 band that was lower. There's a final lane of the gel, which is the DNA from the crime scene, and that lane has 1 band that's lower, and it matches the DNA from suspect F. And so this shows that the DNA found at the scene of the crime matches the DNA of suspect F. So in segment 9, I have asked the students to determine which suspect's DNA matches the DNA from the crime scene. They figure out that it's suspect F. And the lesson ends with a summary of the lesson being about using DNA fingerprinting to tell people apart and how that's used in both paternity testing and forensics analysis. I hope that you enjoy using this lesson, and thanks so much for being a part of Blossoms.